Hey, do you know about microarray? I found that it is a grid of DNA segment of non-sequence printed on a glass microscopic slide.
Yes, it can be used to find out mutation, structural variation, and massive gene expression analysis that occur simultaneously between different cell types. For the example, a healthy cell and cancer cell.
Microarray is very powerful because millions of genes can be analyzed in one single run of the microarray analysis, so we can tell which gene is actively expressed by conducting the microarray.
So how it can be done?
There are different types of array like just spotting the cDNA on the nylon membrane or the glass slide coated with polylysine. Another one is direct oligosynthesis on microarray substrate. The spot on the microarray represents one gene.
The microarray is based on the hybridization of the sample cDNA with immobilized DNA on the chip. Then, the hybridized labeled DNA will emit the light which is visible and detectable for analysis.
Basically, there are 4 steps involved in microarray analysis. There are sample preparation and labeling, hybridization, washing, and image acquisition followed by data analysis.
So the first, we isolate the mRNA. But because mRNA is unstable, it needs to be converted to cDNA, and here reverse transcriptase is used.
The cell can be labeled with fluorescent cyanine dye. For example, normal cell will be labeled with cyanine 3 for the green color, and for the cancer cell will be labeled with cyanine 5 to give red coloration.
So we can apply different cells on different spots to identify which gene is affected, right?
Yes, after that, those unhybridized cDNA must be washed away to prevent false positive results.
Microarray scanner can perform 3 functions, not only to identify color, but also merge the image from red and green scan and show the composite picture. Green is for upregulated, red is for downregulated, while the yellow means that the genes are equally abundant.
Then we can make use of bioinformatic tool to analyze the result very rapidly, especially the gene expression level or the quantity of the gene. But I'm not that clear on what we are going to do with the result.
Well, it depends on your kind of research. It can be used to identify SNPs, which are single nucleotide polymorphisms, which indicate genetic variation that could potentially lead to tumor. Tumor identification markers are developed and can be detected simply by comparing the results between a healthy patient and a cancer patient.
So I guess the cancer can be identified and other gene mutation diseases also can.
Most certainly, using microarray results analysis, comparative genomic hybridization can be done to distinct between different kinds of tumors, such as breast carcinoma, fallopian tube carcinoma, gastric carcinoma, melanoma, lymphoma. We can also study the differences between metastatic and non-metastatic cells. Clark in the year 2000 found that the gene ROC was more present in metastatic cells than non-metastatic cells.
So once we figure out which gene is affected, so this could be aid in the drug development, right?
Exactly, it will be more accurate and reduce the probability of unwanted side effects. Besides that, scientists will have insight to the role of genetics in drug response by comparing the results between non-responders and responders.
Therefore, the microarray assists a lot in pharmacogenomics. The medicine in the future can become more personalized with a better understanding of genes and how they influence the body response to drugs and diseases. Diagnosis of disease is also going to be accurate and less dependent on assumptions that could lead to later decisions.
What any drawback in the microarray?
Yeah, we only can know the presence of the transcript, but we do not know the actual level of the protein, especially our cell does not rely only on the transcriptional regulation. We also have the post-transcriptional, translational, and also post-translational regulation.
Yeah, another problem is the cost. We may require a lot of money to get the result and also to convince the public.
I think the cost will be lower down in future, such as the sequence technique.
Yeah, that's true. I hope it does lower down in the future. Hey, what time now? Hey, we gotta go, let's go. Got class now, let's go. Thanks guys, good discussion guys.
Hey guys, how do you feel about today's lecture for functional genomics? It was incredible, right?
Yeah, finally, after 3 years, everything starts to make sense now, and we are beginning to see the bigger picture where the theory we have studied can be applied in modern medicine.
But I find it rather confusing though. It seems to be a rapidly evolving technology with so many variations, applications. So do you guys want to discuss about this?
Sure, which part do you find most confusing?
Yeah, me too. Do you remember what we learned about EST? I don't quite get it earlier. Can we discuss it now?
Sure, EST is a short fragment of DNA sequence derived from randomly selected clones from the cDNA libraries.
Basically, you can say that EST is part of cDNA. It can be used for the identification of full-length gene and also assist in gene mapping.
Yes, EST is faster and cheaper as compared to full genome sequencing, but most of the steps can be automated. So scientists can use it to provide the experimental evidence for the position of exons, or provide regions coding for potentially new proteins, or even assist in characterization of the alternative splicing.
Yeah, we can use full-length cDNA sequencing and even SNP data mining.
So how is the EST generated?
The mRNA is isolated, followed by reverse transcription in order to synthesize the cDNA. Then the cDNA is then integrated into vector to produce a library.
After that, we use the universal primer to perform single sequencing reaction of 200 to 500 nucleotides at both 5' and 3' ends cDNA. And finally, we examine the quality of EST by using bioinformatic tools and also genomic database to remove those low quality sequence information and also the contaminating vector sequence.
We have to ensure the raw EST are checked for the vector contamination, low complexity and repeat region. Those low quality singleton and very short sequence will be removed.
So it's a readable sequence of a database.
Yes, ESTs are then assembled to generate consensus sequence which are known as the putative transcripts. So DNA database help us to assign, identify homologs, and predict the possible protein functions.
The putative transcripts can be translated into putative peptides, which can be assigned putative functions with protein database similarities such as.
The analysis is extended to functional annotation followed by visualization and also interpretation of the results.
But is there any other function for EST?
Yeah, I remember other than the gene detection and the verification of gene prediction, there are semi-quantitative that allow you to know which gene is abundantly expressed.
Proteomic analysis can also be done with EST.
EST could be used as a bioindicator of exposure of the pollutants and other toxic agents.
Yes, EST is generated from the American oyster, Cassatria virginica, and the earthworm, Lumbricus rubellus, can be used to do so.
The EST provides column for microarray and check for the position of exons.
Wow, so EST is very useful in many fields including the pharmaceutical, the genetics, agricultural, and even the environmental protection.
But then many people may focus on the genomic field, but bioinformatics has also an enormous potential in future. This is because when the genomics tools like the sequencer and thermocycler are getting more and more advanced, huge data can be generated very fast. So we must be able to analyze them without lagging our time.
Yeah, we can make use of EST database as an alternative to library screening or even for cDNA screening.
But they are highly redundant, and databases are skewed for sequences near the 3' end.
Yeah, the error rate is also high in the value EST. They are error-prone and require multiple pre-processing followed by clustering assembly and database matching, functional annotation, yield and yield transcript information.
I think everybody is clear about this topic.
Yeah, it's quite good.
I'm hungry now. Let's
Hey,
go.
let's
Go makan.
eat, guys.
Go lah, go lah, go lah.
Bye-bye.